It's just gone 2 o'clock, so we'll get this webinar underway. Hello, everyone, and welcome to our Mushroom Casing Alternatives Research webinar. My name is Joy Clarke, and I am the Mushroom Research Officer with the Horticulture Development Department at Teagasc. We have 4 great talks lined up for you today. Before we get started, a little bit of housekeeping. So your cameras and your microphones will be switched off for the duration of the webinar. At the bottom of the screen, you'll see a Q&A box. If you have any questions during the webinar, I'd invite you to put your questions in this section. After our 4 speakers have completed their presentations, we'll open a Q&A session where I will direct those questions in that section to our speakers. If your question is for a specific speaker, please include the name of that speaker within the question. Otherwise, we'll open it up to all the panelists. This webinar is being recorded. It will be available shortly on our YouTube channel, so you can watch it back and share it with anyone who may be interested in it. So with that, I'd like to get underway with our first speaker, who is Paul Bishop from Teagasc Mushrooms, and he's going to talk to us about the peat replacement projects that they're conducting. So Paul, if you'd like to share your screen for us, please.
Certainly. Good afternoon.
Thank you. Perfect.
And that should give you the presentation on screen, Joy.
Yeah, we can see that, so you can work away with your presentation, Paul.
Thank you very much indeed, and good afternoon and welcome. My name's Paul from Teagasc Mushrooms in Monaghan, and I'm here to do a presentation on our industrial scale peat replacement project, which has been running from April until October of this year. I'm Paul Bishop. I'm project coordinator with Teagasc Mushrooms, and I've been there for the last 25 years. Teagasc Mushrooms was established in 1999 by Kieran Walsh. We employ some 200 people and produce some 200 tons of fresh white and brown mushrooms per week in 5 farms spread, 4 in County Monaghan and 1 in County Cavan. We specialise in growing breakfast flats for retail, food service and wholesale markets. Our project aim was to replace deep dug peat with non-peat alternatives and productions of mushrooms on a commercial scale. The casing material has been developed by Heartpeat, but not previously tried on a full farm basis. Due to environmental impacts and concerns, the Irish mushroom industry is required to phase out the use of conventional peat as its casing substrate by 2030. This project aims to produce mushrooms on a peat alternative with a reduced carbon footprint. So our target was to produce mushrooms, similar yields and product quality that we were able to produce on deep dug peat. So starting with 1 house of peat-free in March 2025, we gradually stepped up to fill in 2 farms fully with peat-free casing from July 2025. 97 white mushroom crops on our farm in Cookhill East, flats on first and second flushes, and 21 brown mushroom crops on our Kilner Drain farm, portobellos on firsts, second and third flushes of chestnuts. It took us until July of 25 to have full fills on both those 2 farms, and we were filling houses of between 360 metres and 640 square metres of growing area. Our target was to replicate our current mechanical filling operation and to make the growing medium work in our existing environment. So here in these 3 pictures, you can see our casing peat-free replacement. In the very first picture on the left, you can see the material as delivered in the walk-in floor trailer, similar to how our deep dug peat arrives. In the second picture in the middle, you can see our discharge onto the filling belts, and you see us adding water to develop structure similar to the deep dug peat casing. In the third picture on the right-hand side, you can see the use of the spray head lance to ensure a uniform water application being judged by a skilled person. The product mix was too fine, and by adding water to try and make the casing material heavier, increasing the lump size, so that we could use the material in our current mechanical processes. That's the conveyor, the filling, the casing machinery. Working on the stickiness, creating the lump size. We're looking to replicate the deep dug peat. We can recreate the medium or that we already had from deep dug peat. We could use it in our existing systems. We used the casing height in the early days of between 3.7 and 4.5 centimeters, settling on a fill depth of 4 centimeters. The water and the casing inconsistencies caused us issues in the early crops. You can see a picture on the left-hand side of overwatered and on the right, underwatered. We were getting insufficient mycelium growth on the overwatered, too wet a growing medium. It was getting too wet. It was going through the filling process, making its way into houses in clumps. Too wet. Normal watering in the house prevented the mycelium growth from coming through properly, leading to blank areas, no cropping. Right-hand side, underwatered, too dry, too much, an overrun of mycelium, no pins. Our whole process, very sensitive to the normal filling process. Machinery set up, the application rates, the compost. I cannot stress in the whole process how much effort needs to be put into making sure that the house is filled correctly. After 20 years of dealing with deep dug peat, a conclusion is that the fill process has to be correct. There is no second chance to correct it later. We haven't those 20 years of experience that we have in dealing with deep dug peat to rely upon in this new process. We came across competitor mold. It appeared just before pinning. To give our competitor mold its more common name, it's known as cinnamon mold. It caused less mycelium. If the pinning was too wet or too dry for the cacking to stick to, higher temperatures of over 30 degrees at seal up caused it to compete to inhibit pin growth and pin formation. Our competitor mold was tested through micro and residue testing to prove that it was not a safety threat. There was a tendency to overpin in our early trial crops, causing quality issues and reducing picking speeds. These areas were addressed by the growing team by increasing the lump size that was left at the end of day 0, by breaking sooner, darker, not allowing the mycelium as far up as we would have done with our deep dug peat, slowing down the break, keeping CO2 levels up in the houses, and slowing down fan speeds. As you can see from the quality of the product in these 3 pictures, we proved we could get good quality product from this change in growing medium. Our shelf life testing was comparable with our existing cropping. We took the yields of the Clarke farm for 2025 and compared them with the previous year's production of the same site. You can see, therefore, that on firsts, we had a gain of 2%, and on second, a gain of 1%, overall giving us an increase of 3%. Now, this farm is two flush, first on flats, second on flats, and with the farm at full production, we had to go back to compare it to 2024. This is us comparing the Horticultural's production of 2025's period with that for Teagasc's home farm for the crops that produces flats on firsts. And again, you can see that overall in those firsts, we had a 2% increase. Although these yields on this growing medium have seen a slight improvement over the benchmark period of the summer 2024, neither has performed in line with our yield targets for the production sites. Particularly when you look at the first flush on flats, our target would be a benchmark of 24 kilos per meter squared as against the 21. Kiln Drain was our only brown production site in the growing system. So we were able only to compare 2024's against the 2025 production. So on our firsts, a gain of 1, on the seconds, a reduction of 1%, and on the thirds, also a reduction of 1%, giving us an overall reduction of 2%. We were only filling one crop per week and therefore had a limited number of crops during that period of test to base those results upon. It was also the summer growing period, and we had some mechanical issues that were not transparent as much when we were using the slightly more forgiving deep dug peat. We believe the brown production is suitable for this growing media, but requires us to put in more work. Provided full information to Pembroke Alliance to enable comprehensive calculations to be done. The environmental scientist used a standard life cycle adjustment method to quantify the environmental impact over the product's entire life cycle. You can see from the two graphs, the left graph is looking at deep dug peat versus the peat-free casing. We had results resulting between 200 to 250 CO2 per meter cubed for deep dug, and a huge reduction down to 46 to 54 kilograms for CO2 per meter cubed. The right-hand graph is showing us there the footprint of carbon for the mushrooms actually growing on the substrate. For deep dug peat, we had products between the range of 0.37 to 0.46 per kilogram of CO2 against kilograms of mushrooms produced. And then moving on to the peat-free version, we are turning that to 0.09 to 0.1 kilogram of CO2 per kilogram of mushrooms produced. So a big saving. So in summary, our yields on mushroom quality on peat-free casing are on average the same as traditional peat. Although some points to note are the additional costs associated. The yields and the carbon savings. On costs, the major impact on growing the medium is actually 3 to 4 times the cost of deep dug peat. Labour. The additional labour costs of filling an application are necessary to monitor and control the structure of the growing medium. This is a critical control point in the whole process. Yields. We proved we could crop to a comparable yield against deep dug peat, although not in line with our targets. For carbon, over the period of this project, we saved 283 tonnes of CO2. Our future plans. Additional innovation and diversification support will be required for us to better understand the growing medium, to further advance our performance on the peat-free substrate, to continue to reduce the carbon footprint of our business operations, to reduce the labour costs involved in peat-free. We have to do some work on the skilled personnel that we have in place to manage the replication of the growing media, the addition of water. And we need to develop more work on the growing techniques because we've carried out this trial over the summer period and we need to carry on the trial into winter months. I'd like to say thank you very much for watching and listening to our presentation. We'll be happy to answer any questions at the end, and further information will be found in our industrial facts sheet, which we will send out later on in the chat box. So I'd like to say thank you very much.
Thanks very much, Paul, for that excellent presentation. If you want to stop your share screen, if anyone does have questions for Paul, if you could please put them in the Q&A box now, and we'll direct them to him at the end of the session. Thanks, Paul. Thanks very much. So next up, we have Owen Corbett, if you'd like to share your screen, Owen. So Owen is going to talk to us about some of Teagasc's project in the peat alternative area. So Paul, or Owen, take it away. Perfect. I can see your screen there now.
Great. I'll just unmute myself. Thanks, Joy, for that intro. So my name is Owen Corbett. I am a growing media research officer here with Teagasc. And we focus on finding peat alternative growing media across all of the various horticultural sectors in Ireland. And we have a focus on mushrooms in particular due to its importance to the sector as a whole. So today I'm going to be presenting some of our agronomic trial work on peat reduced casings, and particularly taking a look at some of the water dynamics, so the dynamics of hydrology within the casing and how that might affect yield. So I'm going to start with a few introductory concepts on hydrology. So I'll begin here with this glass of water. This glass of water represents a state of free water. And so the molecules within this glass are free to move in any direction that they please, assuming that there's no interactions with the glass itself and there's no dissolved solutes within the water. And assuming as well you're taking your glass of water at sea level. So in this example, there are no external forces acting to establish a water potential gradient. So the pressure potential, which is measured in units of Pascal, is registering as 0 in this case. And organisms have evolved to use the properties of water, so the physical properties of water, to move it as efficiently as possible. So I'm starting with an example of a plant here, but we'll get into mushrooms. And I'm using a plant because they're really well studied and understood in terms of how they move water. So in a process called evapotranspirations, plants can efficiently draw an unbroken chain of water molecules in at their roots and control this loss of water through their leaves, and in effect operate some control over the transport of water and the nutrients dissolved within that water. And so effectively they're moving water across a water potential gradient from areas of high relative potential to areas of low relative potential or energy potential. So because plants can be dissected and examined a lot more easily than mushrooms, the journey from root to tip of this water is very well understood. So there are points along this journey where the pressures have been decided and are known. However, these are not hard and fast values because in reality, there are plenty of influencing factors on the water potential. And so I'm going to go through some of these in turn as they relate to the mushroom crop. So water is attracted to most surfaces. And so in this example, they are attracted through hydrogen bonding and weak electrical forces to the particulate matter in soil. So the strength of that attraction is known as the matrix potential, and it's measured in units of pressure as well. So generally speaking, in this example of soil, it's negative 300 kilopascals, so kPa. And this is the availability of water to the plant. It relies on the soil-contained water having a higher water potential relative to that of the water inside the roots. So if that's the case, water is drawn into the roots passively. So the plants don't have to expend any energy in order to draw in water. But as the material dries, so in the example here, maybe in drought conditions, air infiltrates the porosity, replacing water. And so whatever water remains is much more tightly bound to the particles within the soil. And so the plants have to then expend cellular energy in order to draw that water out and break those bonds loose. And in fact, at a certain point, at negative 1500 kPa, most plants experience what's called permanent wilting point. Effectively, they die. They're no longer able to generate sufficient pulling force on that water in order to extract it. Another aspect of water translocation is the process of osmosis, which is regulated by the concentration of solutes within the water. So in this example here, we have a high solute concentration within the roots of a plant, and that's able to passively draw water in across a selectively permeable membrane, as you probably remember from your school days. But if the gradient is in the opposite direction, water is then pulled in that direction. So both plants and indeed mushrooms can experience osmotic salt stress if the immediate environment outside of their cell structure is more concentrated in solutes. So there are other factors too which influence the transport of water, and we'll move on to those in terms of how they relate to the mushroom system. So unlike plants, the commercial mushroom crop doesn't have to transport water across great distances, like in the example here of the giant sequoia trees. They have to overcome gravitational potential. So we can assume it's not very relevant for the mushroom system, which is only an order of centimeters high. But what is important is humidity. And so within the growing room of a mushroom crop, we're able to control the relative humidity. And so the mushrooms are incredibly efficient, more so than plants, at the evaporative loss of water. And that's actually how they drive, in part, the water movement and the nutrient movement into mushrooms as they develop. And in nature, fungi operate a kind of a ratcheting system. So by day, when the temperatures are warm, they are efficiently evaporating water from their surface. And in so doing, they're able to concentrate solutes inside their mycelial structure. During the night, when temperatures are relatively lower, evaporative loss of water is also slower, and they're able to use the solutes that they concentrated during the day in order to pump water effectively into developing mushrooms. And so that's why often at night you'll see mushrooms appearing and expanding. So in short, Agaricus are influenced greatly by matrix potential and to a lesser extent by osmotic potential too. So that's why it's important to keep casing at a EC below, say, 1500 millisiemens, because they are sensitive to salt stress. So to get to some of the work that we did in Teagasc, we looked at amending peat-based casing with wood-based material. So amending them with wood fiber, uncomposted barks, and composted barks. And we did this in volume proportion with peat at 30, 50, and 70%. And our control in each case was an industry standard wet dug peat casing. So in terms of water content, this is often cited as an important thing to monitor. And so between trials, if you look at the two differing circles there, we're within uncertainty between those two trials of the gravimetric water content each time, which means we did a good job at saturating the media consistently in both trials. Peat, generally speaking, on day 0, as it's going on the crop, is about 84 to 85% gravimetric water content. And what we see in the wood fibers is that with an increasing proportional addition of wood fiber up to 70% by volume, there's an increased gravimetric water content up to 88 or 89%. We see the opposite trend in the barks, however. So with an increasing component of bark, uncomposted or composted, we see a stepwise reduction in the volumetric water content, or the gravimetric water content of these media. But water content is not the be-all and end-all. What's important is the ability of these casing media to release that water as mushroom demand increases. So just to explain what you're seeing on the screen here a little bit, we have a unit in the lab called a HyProp2, and this is capable of plotting volumetric water content versus matrix potential on the y-axis. And so the curves that you see on screen are called characteristic release curves. These are characteristic for a given media type. So regardless of the crop conditions, these characteristics stay consistent. So what you see there is that between 80 and 90% by volume of water, there's plenty of availability. So it's a little bit like the glass of water at the start. The matrix potential is at 0 pascals. But as the media dries downwards, so towards the 10% volumetric range, you see that there's a much lower matrix potential. So organisms are going to have to work a lot harder in order to extract water as media dries like this. And so we can explore this in a little bit more detail later on, but that's some context. So the Teagasc Mushroom Research Facility, some of you may be familiar. Just to say, we're able to fill at a commercial rate into these grey Euro crates, and that gives us the ability to generate a complete randomized block design within the shelving system of the research facility. So to yield, just to explain the graph a little bit, the colored portion of the columns that you see there represent Class A graded mushrooms. So good quality mushrooms, nice and white, while the stacked portion on top in white are the proportion of Class B graded mushrooms. So B graded mushrooms are on the whole generally classed down due to symptoms of water stress. So premature opening of the veil, split stipes for the most part. Some others were stained or had dirty caps as well, but to a lesser extent. So the wood fiber treatments in yellow are statistically similar to peat in terms of their yielding efficiency of Class A graded mushrooms. The uncomposted barks there in green show a stepwise reduction in yield, so significantly less yield in Class A mushrooms to peat. And the composted based casings in brown there differed slightly in terms of their trial performance. So in trial 1, we see a similar stepwise reduction in Class A graded mushrooms like we saw in the uncomposted barks, with a high proportion of Class B graded mushrooms. While in the second iteration of the trial, we see an improvement of Class A yields, and we can discuss maybe why this is a little later on. So in terms of how we monitored the performance of the casing hydrology, we installed 2 sensors in the center of the casing, a moisture sensor and a tensiometer. And what the tensiometer is effectively a moisture sensor which is encapsulated with a ceramic coating, which is a porous media that's really well characterized and understood in terms of how water moves across the porous membrane. And so as the casing dries, it's in equilibrium with the tensiometer, and the water within that ceramic encapsulation is pulled on, and it's the force of displacement of water with air that's recorded by the computer there. So we're recording those every 10 minutes throughout the course of a crop. So in terms of what this looks like in results, that bottom section of the graph there in red is the volumetric water content trend as measured by the moisture sensor through time. And generally speaking, what we see is at the beginning, day 0, we have a saturated condition, like that glass of water we discussed at the start. So for the peak control here in black, that's 85% moisture content, which is generally trending down over the course of 35 days of the crop. The black arrows indicate watering events. So after each flush, which are in those gray shaded regions, we water the crop, and you can see that the moisture sensor picks those up. Then, as we look at the tensiometer data, which is on the top half of the graph there, so this is measuring that matrix potential, which I discussed earlier. And so as the media dries within a flush, the tensiometer is able to register a negative excursion in this pressure potential as water is used up. So there's very good correlation between the use of water during a flush, which is those gray shaded regions, and the onset of matrix potential reduction. So things get ever more complicated, I'm afraid, but overlaid on the black trace of the peat control casing, I've now put in our wood fiber treatments. And as you can see, they're quite well correlated to the peat-based control in terms of their shape, timing, and magnitude. And remember, they were statistically similar in terms of their yield as well. So if we look back to some of the characteristic curves, this makes sense, because what we see here in the green shaded region is sort of the operational zone of the mushrooms, if you like. The lowest magnitude matrix potential I've seen in the literature is about negative 130 kPa. And as you can see, within that operational range, if you like, for both wood fibers and peat, they are behaving quite similarly in their ability to release water to the crop. What you can see outside of this zone, however, is that the two lines begin to diverge. But just to note that the very end point on the y-axis there is atmosphere. So there's no case that mushrooms are ever going to be able to exert that much force on water. So what's key is that sort of operational range of the mushroom crop. So when we look at some of the other treatments then, the uncomposted barks, the most notable thing is a shift in crop timing, so a delay. So at the top of the graph there, you might be able to make out there's dark gray kind of bookmarks. They are highlighting the regions where both peat and wood fibers were flushing, as I showed you earlier. But you can now see there's an offset towards a later cropping time for the uncomposted barks. And with an increasing proportion of uncomposted bark within a mix, the effect gets more pronounced. So the 70% barks are flushing up to 4 days later than the peat control. And you can see this very well in this picture, which is taken on the first day of flush 1. And you can see there on the top row, the peat-based casing and the wood fiber treatments are flushing in a similar fashion, whereas if we look at the uncomposted barks there in the middle row, you can see that there's a delay, and that delay is a stepwise delay, like I mentioned. Now to the composted barks, I suppose the most notable thing is the magnitude of that suction pressure that's occurring in the first flush. So generally, bark media were much more loosely structured than peat, and the airspace within that structure, I believe, is the reason for their propensity for overpinning, which was observed in these crops. So when the crop overpins in that first flush, it tended to generate this high magnitude drawdown of water. That means then in the second flush, it leads to a complete use up of all the available water within the crop, as you can see. On the bed, the crop is stalling, so that's observed on the bed. So mushrooms have slowed or are no longer expanding, and no new mushrooms are forming. And this correlates with observations in the data from the matrix potential tensiometers, where this sort of plateau emerges in the curve. So you've run out of that available water, and the crop begins to stall. And that usually occurred after high magnitude drawdown of overpinned first flushes. So if we try and diagnose then what we see in terms of matrix responses as measured by the tensiometers, when the crop is progressing well and mushrooms are developing with good stagger, we have a healthy growth that's characterized by this slightly convex curve with a flat back. So it's a little bit like a blade shape. Along the back of the blade there is where we have watered the crop, and the matrix potential goes right back up to that state of free water, and is available then to the next flush. And the media is completely saturated once again. Where the development of mushrooms is slow or slower, we see a slightly shallow concave shape to the developing curve, signifying less available water. And often these flushes are much more protracted in duration as well. And like we saw in the final example of the composted barks, we have a case of stalled growth. So there's no mushrooms forming on the bed, no new mushrooms forming, and whatever's there is slow to expand. So there's very little or no available water. So we see this plateau effect. So I suppose the question that's raised then is, can we use tensiometers even to predict yields? So if we look at this through measuring various metrics derived from the curve shape as it evolves through time, I looked at duration to see if it had any correlation with yield. We also looked at minimum pressure achieved. So that was the lowest value recorded in the matrix potential curves, and also looking at the mean pressure value over the course of a flush. So to investigate the predictive capacity of these 4 different metrics, I looked at the residual yield. So I've taken out the confounding factor of flush-wise decrease in yields that you generally see from first flush to third. And we can interrogate aspects of the matrix curve a little bit better when we remove that confounding factor. And we didn't see any predictive capacity or strong correlation between yield and minimum or mean pressures. So I calculated what we call a full width half minimum value, which is the length of time that the minimum pressure is below the halfway point. And in real terms, that's what it looks like on measurements, live measurements of the crop. So for the wood fibers that performed well in terms of their yields, you can see the curves are similar in shape and duration, even across the 2 replicate trials, so the red and the black traces there. By comparison, the uncomposted barks, the 3 graphs along the bottom, what we found they didn't do so well in terms of yield, and they have a divergence in that full width half maximum value between trials. So in this example, you can particularly see where that value is able to distinguish between casing layers that have exhausted their available water and where you ended up with the plateauing of that matrix potential. So if you look at the relationship to residual yield on this full width half maximum value in the first trial, with the bark-based casings which ran out of that available water, if I just circle the examples there that showed the plateau, these all perform poorly in terms of their residual yields. And this is what they look like in terms of their those full width half maximum values when they're stacked together. So in extreme cases where there's a severe lack of water in casing media, yes, the full width half maximum values are useful, as too is duration as well. But more work is needed to improve the predictive modeling. So we can show that tensiometers have major potential as diagnostic tools for observing patterns of mushroom growth. So for instance, if the crop stagger isn't good, when there's overpinning or when yield is down. But this needs further work, I think, in future. So in conclusion, in terms of agronomic work, at least in terms of the wood fibers, they performed very similarly in terms of their matrix potential to that of peat, and that underpinned their performance in terms of yield as well. So they performed non-significantly different from peat. While the uncomposted barks exhibited significantly diminished yields compared to peat, they also had a cropping delay, which is a concern. They had a loose structure and a propensity for overpinning. And what we found was in the second iteration of the trial, where the material was held for 3 months, we also had an infestation of cessid fly larvae with the uncomposted bark. So that renders it high-risk media, if not an unusable one. For the composted barks, there was a difference in terms of the yielding performance across 2 trials, and we put that down to a different agronomic approach that was taken in the second trial, in which we extended the case run period for 2 days to allow this looser structure to colonize better, and that improved performance, or at least reduces the class B yielded mushrooms for the composted barks, and they became comparable to peat then in the second trial. It also reduced the cropping delay as well. And so often the challenge with bark, generally speaking, and not so much in this trial because we use the same parent material, but is getting the same consistency with each batch in terms of the supply of bark. And what's critically important for compost bark into the future, if you're using it, is to understand the stability of the material itself. So ensuring you have a really well composted material is important. And as I mentioned before, tensiometers are potentially useful diagnostic tools, and they may potentially guide agronomic decision-making into the future. But further modeling and understanding of these prediction models is required for that. So with that, I'll say thank you. And we do have a publication on this, which is open access for anyone who wishes to read it. So there's a QR code there on screen if you want to read in more detail. So Joy, over to you. Thank you very much.
Great, thanks, Owen. And that publication is in our latest newsletter as well, so you can find it there also. And great, so thank you, Owen, very much. You can stop sharing your screen now, and we'll ask Nancy to share her screen. So Nancy Pick is our next speaker. She's from Inagro, and she's going to talk to us about some EU projects looking at peat alternatives, which is peatless and biochamp as well. Perfect, so great. Great, we can see your screen now, so you can go ahead.
Perfect. Yes, hello, good afternoon, everybody. Thank you, Joy, for this kind introduction. And thank you also, Ev and Teagasc, yes, for this invitation to join this interesting webinar on casing alternatives. So really happy to join. So today I would like to talk about the project Biochamp we have been working on for the last 4 years together with other partners, and also the recent launch of a new project called Peatless. So the goal of Biochamp was to develop an alternative and sustainable low peat casing and a biostimulant for the mushroom industry in order to reduce the need for pesticides and to contribute to improving the productivity, sustainability, and the profitability of the European mushroom sector. In the project Biochamp, in total 13 partners were involved, coming from 7 countries: Belgium, Spain, the Netherlands, Serbia, Poland, United Kingdom, and Italy. It was a project that was going for 4 years, so it ended last year, at the autumn last year. And the partners were a mix of research institutions, like Inagro and Teagasc. That was a project coordinator from Spain. Also, University of Wageningen and Oxford were involved. Also, University of Italy. Also, professional mushroom growers were partner, next to some industry partners like Newfos and Kikila BBB. This flow shows the work plan of the practical work we have done in the project. So on the one hand, we started with small-scale trials, small-scale crop trials, using crats of 1.2 square meter, like the one Owen just mentioned, also in Teagasc. This was a trial. These were the trials where we are exploring several alternative casing materials. And next to this, we were also working on microbiota to find ways how they could help us in order to further optimize the peat-less casing materials. And the next step, scaling up, was done of the trials in both at Inagro and Teagasc, to come to a very important stage, which was the validation of the products on a commercial scale. And that was done at the professional mushroom growers that were part of the project, to come finally to the commercial strategy. So starting off with the small scale trials, we have selected our main peat alternatives materials based on earlier work from the group of Jan van der Wolf of University of Wageningen in the Netherlands, work done by Tanvi Taparia, and we focus mainly on 2 types of grass A and B, sphagnum moss, spent casing, and green compost. And we used peat as a traditional peat as control. We tested different mixes, and we replaced up to 75% of the peat in our crops. We tested also steamed and unsteamed material, and we tested different parameters like yield, quality, and disease incidence of the different alternative casing source. Just to give an idea of what material we are talking about, one material was the grass fibers, the fermented grass fibers that were provided by the firm Nufos. It's a Dutch firm, and those fibers are produced by a circular biorefining process that converts non-woody biomass into ligneous cellulosic fibers. The next material was sphagnum moss, provided by Kukila BVB. It contains non-decomposed sphagnum species, and the sphagnum moss contains different species, so it's a complex of different sphagnum moss species. It's grown on degraded peatlands, and that's at the same time a big advantage because it can be harvested repeatedly and sustainable. Next product was a spent casing soil. It's like we all know, it's mechanically cooked out. After cookout of the growing rooms at the end of each cropping cycle. And then there's also green compost, which is in this current study in the Project Biochamp, was made of autumn leaves of various tree species. And it was processed through aerobic composting. So next to the alternative peat materials, we were working on biostimulants. We have been selecting most promising bacteria, and there were different strains of Pseudomonas putida fluorescens and Bacillus felicensis. We tested the effects of those microbiota to see if it would have a positive effect on yield, and also if they could play, let's say, a positive role in crop protection management. So to see if they would give any effect against particular crop pathogens like dry bubble, cobweb, or red bubble. A specific Tachman assay was designed to quantify the presence of the biostimulants, and the goal was to select the most appropriate doses and the most suitable timing for the application during the crop. So in other words, was it possible to use microbiology to further optimize our alternative peat materials? So 7 types of casing soils were tested: grass, green compost, spent casing, sphagnum moss, and peat, and also in different mixes. And next to this, several mixes of the biostimulants were tested, and also in different doses. Here are some impressions of a practical work. So these are all photographs from trials at Inaro. You see on the right figure, given the different colors of the casing material of each grade, you can see there are different casing material that is applied. Here are some pictures of the growing rooms at Teagasc in Spain. So to come back to this flow of the work plan, research showed from the many practical trials we did that 2 alternatives showed the most potential. It was the grass and the sphagnum moss, each in a 50% part. And looking at the biostimulants, we saw that the most potential microbiota was the Bacillus felicensis TCM19 that had the best effect. So in the next stage, scaling up was done. We did some semi-commercial trials, again at Inaro at Teach. We selected, we focused on the 2 alternatives, the grass and the sphagnum moss, using the traditional peat as control and applying the Bacillus felicensis strain. Here is a impression of such trial at a larger scale, and no longer the small crates from 0.2 square meter, but each tray here has a surface of 1.5 square meter. Some practical work to mix the casing alternatives. These are some pictures from Teagasc, Spain, and also from their growing rooms. So next to the work on the alternative materials, extra work was also done on the part for the biostimulants. Many casing samples have been collected and analyzed with Tachman assay. And next to this, also a lot of amplicon sequencing and PLFA analysis were done to have a better idea of the population dynamics of the applied bacillus. So to come back again to this flow and to communicate the results, despite many trials we have done for testing the effect of biostimulants, unfortunately, those trials did not show any positive effects on the yields of these trials. But the good news is that despite the negative effect of the biostimulants, both selected alternative materials, the grass and the sphagnum moss, still showed good potential to take them into further trials. So the next step was a very important step at the cropping trials at the commercial scale. So at the commercial mushroom production sites. There were 4 in total, one in Spain, Eurochamp, one in was Ecofungi in Serbia, a production site from Kikila BVB in the Netherlands, and a big production site in Poland, the Kiwala mushroom site. So this is just to give an impression of the grower site in of Poland and the Netherlands. They used let's say the traditional Dutch system, the phase 3 in bulk, automatic control system, also automatic irrigation system. In the Netherlands and in Poland, 3 trials were done in total. This is a picture of the site in Serbia, which is a more traditional system, let's say. It was also an organic cultivation system. No phase 3 in bulk, but phase 2 in bags were used. They make their own compost, irrigated by hand, so a big difference. And they have performed 12 trials in total with the alternative materials. And this is a picture from the plant in Spain, Eurochamp. They use phase 2 in bags, also irrigated by hand, and also they have performed 3 trials in total. So during those trials, the feedback of the commercial mushroom growers was very, very important for us. And this here is a short list of some of the main points they found that were important to take into account when working with those alternative materials. They monitored there was no need for an additional step for steaming for alternative casing soils. So that's a quite economic benefit. Pre-wetting must be carefully assessed, and also extra attention must be paid to the watering regime during cultivation, mainly because the grass-based casing soils have a lower water holding capacity. I was surprised to hear in the talk of the first speaker from Paul Bishop that also he mentioned that pre-wetting is a very crucial thing. So that's something that we also monitored during those commercial trials. Looking at yields and mushroom quality, both were very promising when comparing the traditional peat and the tested alternatives, and also crop control and management was comparable between the alternative casing soils and the standard casing soil. So to come back to the flow of the work plan, we were very happy that both alternative casing showed indeed potential for further exploration. So the next steps were taken to further commercialize the strategy, but that's something I will not go into deeper in this presentation because my time is too limited. So yeah. Next to the cultivation trials, a lot of work has been done, especially by the people from the Italian university, from the Italian partner, to work on life cycle assessments, how to evaluate, which evaluates the environmental impacts of a product across its entire life cycle, so from beginning to end, let's say. We have published 2 articles on LCA analysis. So for people who are interested, they are more than welcome to take a look at those. It's not easy to summarize the work of 4 years into this presentation. So for people who are curious, I would like to invite them to take a look at the Biochamp websites, where you can find multiple information sheets or posters or other articles or practice abstracts. So yeah, feel free to take a look in them. And so next to Biochamp, like a kind of continuation of the project, I would like to introduce a new project, Peatless. It recently started in May this year. It's also a youth-funded project for 2.5 years. It's also coordinated by Stitch to Spain Institute as Biochamp. 4 countries are involved, in total 12 partners. And this time not only mushrooms or focus group, but also seedlings and ornamentals. So both in Spain and Belgium, mushroom cultivation is one of the focus crops. So yeah, these are then the 2 main countries doing the mushrooms research. Like I just mentioned, there are 3 key horticultural systems, mushroom seedlings and ornamentals, that are studied in Peatless across the 4 major horticulture areas in Europe. And yeah, the ultimate goal is to lower the carbon footprint of horticulture, supporting the green transition and accelerating the sustainable transformation of the total agri-food sector. As in Biochamp, we will define alternative substrates because there are still a lot of challenges to take that are remaining, not only for mushroom cultivation, but also for ornamentals and vegetable seedling production. So in Peatless, we see 3 kind of barriers or hurdles to overcome: technical, sociocultural, and economic barriers. Technical barriers are maybe the most obvious, let's say, because also till now there's no single material, no single alternative that can already match the large demand for peat. Regarding sociocultural barriers, growers are concerned about the substrates that are now available, the alternative substrates that are now available. They are not so robust as a peat, or they need more support and more advice. And then there's also an important economic hurdle. I think we all feel that there will be higher initial costs involved for growers and substrate producers. And there's also insufficient government support and lack of certain incentives. In peatless, we will try to resolve those barriers by several approaches. So going to the first one on a technical matter, we will try to develop many alternative substrate blends with a maximum of 30% peat in all 3 cropping regimes involved. Regarding social culture, we will try to enhance the confidence to make the adoption smoother of the peat-free alternatives. We will give also seminars, provide other educational materials so we can try to help the growers and the substrate producers. And we will also try to establish new business model to promote the market entry of the peat-free substrates. So this was in a nutshell the results of BioChamp and an introduction to peatless. And regarding all the work that has been done at Teagasc in BioChamp, yes, I want to thank the whole team of edible mushrooms. And if there were any questions, I will be happy to answer them at the end of the session. So thank you.
Thanks so much, Nancy. That was fantastic and really interesting to learn about those projects. If anyone has questions for Nancy again, please pop them in the Q&A box now and we'll ask them after our last speaker, which is Christoph Jeesens, who's also from Inagro. And he's going to talk to us about his Alter Peat project. So we can get your screen up, Christoph. Perfect. I can see that full screen. Yep, that's perfect.
Okay, thank you, Joy. Hello, everybody. Good afternoon. I will tell you something about the project Alter Peat and the ongoing trials where we explore peat alternatives in mushroom cultivation. This project is funded by the Flemish government. It's a FLYO-LA project, and the timing of the project is from November 2023 until November 2027. The main objective of this project is a transition towards peat-reduced cultivation substrates without compromising the competitiveness of the primary sector. We have some specific objectives that are involved in this project. In the first step, we want to make an inventory, and we develop an Alter Peat matrix with all the raw materials involved. In the second phase, we want to develop and optimize the peat-reduced substrates. And in the third phase, we want to do demonstration with the peat-reduced cultivation substrates. This project involves ornamental crops, strawberries, woody small fruits, and also mushrooms. We do not do this project on our own. We have a project steering group consisting of partners, co-financers, and observators. The partners in this project are Inagro, which has the project lead, and we also have the research on mushrooms and strawberries. We have Ilvo partner who does the analysis and some optimization. Via Verda is a research center that is involved for research on the ornamental crops. And we have Proefcentrum Hoogstraat, which also does research on strawberries. And PC Fruit is a research facility for the woody small fruits. The co-financers consist mainly of suppliers of substrates, biomass, additives, or sensors. And our observators in this steering group are mainly growers per cultivation group, also some consultants and some government people. Going back now to the first specific objective in this project, namely the making of an inventory of possible peat substitutes. There are 2 steps we follow. In step 1, the peat substitutes are subjected to a physical-chemical analysis to characterize the materials. And based on the analysis results, these different characteristics receive a score, or a score system is applied. to generate a score for suitability as a peat substitute. The score points go up to a maximum of 16, which predict good suitability. And this scoring system for suitability is in fact a result from another project, and it is the basics for peat substitutes in potting soil. So not yet for mushrooms, but it's more for potting soil. The score system is based on pH, conductivity, organic matter, nutrients, inorganic carbon, oxygen uptake rate, nitrogen immobilization, and bulk density parameters. All these analyses together make a score for a suitability of a product. So this score system should help to predict peat replacement suitability in a fast way. So before doing trials. In this project, score system is used to help researchers make choices for their trials. And it's also used rather as a tool than an objective. Now, together with the casing soil suppliers, we try to translate the scoring system for potting soil to peat substitutes to an applicable version for casing soil peat substitutes, so for mushrooms. Because in casing soil for mushrooms, we have other restrictions than peat substitutes in potting soil. Hereby, I share you the results of the brainstorm. From the left to the right, we have a column with the properties that are important, the units, and then the scores that we can give. Scores can go up from 0 to 2, but there's also a possibility to have a -1 score if the parameter is really not good. For water absorption capacity and water holding capacity, there is no or not enough budget in this project to test all the materials, all the screened materials. So current score for mushrooms is not fully covered by all the analyses we actually want to make a good score. Okay, that was a setup of the first specific objective in this project. I will come back to the results of the screening in a further slide. But first, I want to talk you through the second objective now, namely the development and optimization of the peat-reduced casing soils. What you see here is the planning of the trials with the casing soil in this project, the trials on mushrooms. Where are we now? So now we are at the end of the second year, and so far we have executed 3 trials. Next trial is planned in December, and next year it is also planned to scale up the cultivation surface per tub from 0.2 square meters to tubs of 1.5 square meters. So if I talk some about the trial setup of the first trials, we have tested 46 potential peat substitutes in the 0.2 square meter tubs in 4 parallels. The peat substitutes were mixed with standard peat casing soil in a 50% or a 30% mix, of which 30% is the alternative and 70% peat or peat casing soil. Mostly if analysis showed high conductivity or presence of ammonia or a low stability, we mixed only 30% of the alternative in the mix. And we conducted 3 flushes per trial. Hereby some pictures of the trial setup. So we collected all the materials we wanted to test. We weighed them and put them separately. We pre-wetted all of the alternative casings. We mixed it together with peat casing, and we covered our cases or our tubs. The tubs were put in a cultivation room, and we started the cultivation of the mushrooms. So we had here the pinning, and then we had the further trial ongoing, first flush. As we have tested 46 materials, we have also made a lot of observations about the working points that need to be improved before we can use the peat substitute in the casing soil. Hereby, I want to share with you some pictures as example of what we are talking about. So one thing that we sometimes see was a sealed surface. I don't know if that's the correct translation in English, but on the left side, you see that the casing soil is a bit collapsing if you give water. That's something we do not really want because that will affect the mycelium growth. Other things that we observed were infections during the mycelium growth. We also had mycelium growth inhibitions. We saw other infections during first flush, so other mushrooms or other fungi. We had infections with blotch, bacterial blotch, or also on the right side, you see some dirty mushrooms or some pieces of alternative casing on the mushrooms. That's also something that we do not want. We had lower yields. We observed trichoderma infections. Luckily, we also observed some good alternatives that we can start working about. All these 46 materials we tried to, all these observations we tried to score, and we had measured and we tried to visualize in a result table, which is used as a basics for developing further trials. Also, the analysis results from the screening phase, remember the matrix, were added in this table, and color codes are used for better visualization because we are talking about 46 different materials. If you then try to go back to the matrix scores, and we want to compare these scores with the trial results, the overall color code per material. So green means overall positive evaluation of the trial observations. Unfortunately, we must acknowledge that there is no direct correlation between the matrix score and the trial observations. And so not all positive matrix scores of, for example, 12 resulted in a very good trial. And like a direct correlation would show all greens on top and all reds on below of this table. It means actually for mushrooms, the matrix score system should be optimized. So we are not there yet. Talking about results, I'll try to, for the trial results, I've tried to summarize some results per category of peat substitute. Of course, between the categories, there are differences within the category between different raw materials, but generally I've tried to summarize the working points. So for wood, fibers, bark, we've seen that these materials are vulnerable for infections with green molds or blotch. We also saw inhibition of mycelial growth and pinning, probably due to wood species or the infections, and we had moderate yield results. The digestates we tested mostly had a presence of ammonia and low stability. That's why we only mixed 30% of them. They were also vulnerable for infections, and we also saw inhibition of mycelial growth and pinning, probably due to the presence of ammonia. So we also had average yields, or we had average yields. When you looked at composts, that could be bark or green composts, are also vulnerable for infections with green molds or other fungi. They are vulnerable for the sealed surface, vulnerable for contamination with dirt on the mushrooms. And we also saw some inhibition of mycelial growth and pinning, probably due to infections. And they generated average yields. Plant fibers, also vulnerable for infections, green molds, other fungi or blotch, vulnerable for sealing surface. We also saw inhibition of mycelial growth, probably due to the infections, and gave average yields. We also tested woody biochars. We didn't have any infections. They're a bit vulnerable to dirt on the mushrooms, and actually we had good yields on that. If we go to coconut and coconut derivatives, we didn't see any infections, but we saw lower pinning and low yields. I also tested spent mushroom substrate, which is the mix of casing and compost, and it was composted. It had a bit presence of ammonia, high conductivity, so we mixed it 30%. We had some inhibition of mycelial growth and pinning, probably due to the high conductivity or even the presence of ammonia. It generated average yields. Another group, the processed manure that we tested, had a bit presence of ammonia, low stability, so we mixed 30%. We didn't see infections or diseases. It was vulnerable for sealing surface. We saw some inhibition of mycelial growth and pinning, probably due to the ammonia, and it generated average yields. We also tested spent strawberry growing medium. We didn't see infections and no inhibitions. It is vulnerable for sealing surface and vulnerable for contamination, dirt on the mushrooms, but it generated also good yields. And finally, we also tested sphagnum moss, didn't give infections, vulnerable for sealing surface, vulnerable for contamination on the mushrooms, and it generated average yields. Okay, that was a summary of about 46 materials. So this is where we are today in this project. Next step is trying to optimize the better performing raw materials. So what is our plan? We want to add lime or select lime to the blends, because we had some positive small scale trials with it, and we hope to buffer pH in a better way and to lower the amount of infections. We also will add biochar as a component to give extra structure, to hope to have less collapsing surface. And we also already saw positive effects in small scale trials with biochar. We also will add some hydrogel granules to the plant fibers and wood fibers that we continue with to improve the water holding capacity. And also important is that good performing peat substitutes need to be confirmed, because today we only have 2 trials, so we need to do more trials and more confirmation to go further on. This is my final slide. So if you want more information about this project, you can follow our website, or you can hashtag AlterPeat via LinkedIn, or we will disseminate also via other webinars or congresses. If you have information for me about additives that I can use to improve the stickiness or structure, or if you have other experiences with peat substitutes in casing soil, please do not hesitate to inform me. Here's my email address. And I thank you for your attention, and I am happy to answer your questions if you have any. I'll now stop sharing.
Great, thanks, Christoph. That was fantastic, very comprehensive, lots of materials to test. So thank you to everyone for their presentations. We'll now move on to the Q&A portion of our webinar. So again, there is a Q&A section if anyone wants to drop a question in there. Our first question that we have come in is for Paul from James. Paul, James wants to know what units is used in the peat-free. So is it sold in? Is it by cubic meter or by ton? And then also, did you see any difference in pick rates or the cost of picking between the 2 peat types?
Well, in answer to question number 1, first of all, it comes to us in cubic meters. So that's how it's sold and transported to us. And then in the early crops, especially when we talked about overpinning, etc., yes, of course, we saw some change in our yield and, more importantly, in our picking costs, because we had more work to do, or the great pickers had more work to do in getting that product off the beds, too closely pinned together, a lot more work, a lot more baby buttons. So yes, increased costs.
Great. And I've just shared that link there, Paul, if you want to describe what that is for everyone.
The link will take you through to our fact sheet, which gives you the detailed bullet pointers on the whole project over its period. So if anyone wants to click on that link, that will take you through to that fact sheet.
Perfect. Thanks, Paul. Great. So no more questions in currently. I guess I'll ask a question and I'll open it up to everyone here. I guess in your opinion, what kind of effect is the Irish regulations and the EU regulations having on the uptake of peat alternatives in the mushroom sector? And anyone can answer that if they'd like.
So Joy, just to clarify, do you mean regulations around the extraction and use of peat?
Yeah, yeah.
That's a very difficult one to answer. But my feeling is more so than policy driving, the shift to alternatives is the demand from the multiples and the supermarkets may push the boat out in terms of moving the dial towards peat-free and peat alternative materials. There is currently, as far as I know, no policy in place yet in Ireland to limit or restrict the use of peat in terms of a concrete timeline. That hasn't been decided yet. It's different, I know, in the Netherlands. So
Great.
And it would be fair to say that we would agree with that particular position, is that at the moment we would see pressure come in, or we would expect to see pressure come in from the retailers to move us in that direction rather than us being sort of shoved in that direction at the moment.
Excellent. Great. Thank you. And I guess a few of your talks mentioned the likes of mold and trichoderma popping up a bit more. So do you think that's something that we'll have to maybe shift our focus to in the future, or will it have a big impact if we bring in these peat-free or peat alternatives?
Yes, I think so. I think if we have issues with other molds, we should avoid at any time. So in altered meat, also we try to focus on the materials who do not generate that kind of molds, because it's not easy to solve if they are there, or you need to do some hygienization or something, but that will cost a lot more. And there are alternatives who do not show it or do not have it. Yeah, but also the peat reference can have trichoderma, so it's not always that clear or that easy, but we try to focus on the the materials that do not generate diseases.
yeah, I think in terms of the steaming of materials as well, I'm not sure that there's any benefit to doing it in terms of reducing competitor mold incidents. Sometimes, in fact, you can increase the abundance of mold by steaming a material, because you're effectively opening it up to anything that wants to colonize it. So I think molds are a huge challenge, and I think there's more work needs to be done in terms of understanding the impact of molds on the crop as well. So potentially some molds might not affect, although they're unsightly, they might not affect the yield of mushrooms, and I think more work needs to be done on that point.
Great.
In Biochamp, we have tested steamed and unsteamed material, and we did not see any differences. So that means no need to steam material. And I think it was in the presentation of Paul also that he mentioned that there was some infection of Chromosporium, like the Pezisa caps, the satellite caps, let's say. We also saw that on the material in Biochamp, but we did not see any effect on the yield. So yeah, we don't see it as a real competitor mold.
Mm-hmm.
I think often with the surface molds you get, at least the small ones, they disappear soon after watering as well. So I don't know, again, more work needs to be done in terms of what effect watering back these molds would have in terms of their spread. It's not something that we've done, but we have noticed the occurrence and then the knockback effect of watering over molds as well.
Certainly with the work that we, certainly with the work.
You're muted there, Paul.
Yeah, we just lost you there, Paul, if you unmute.
Do apologize, is that if you go back to the work that we've done on trying to make our growing media very similar in structure to our peat casing, yes, is our opinion at the moment would be is we're not looking to expect to see any more or much more competition than we've already got. So we're not at the moment looking to say, well, we're going to get an increased amount of competition or mold than we would have through our current growing system. But that's perhaps because we're looking to make our substrate very similar to the way our deep dug peat is at the moment, and we'll keep our growing practices the same. But just like everyone said, there's a lot of work still to be done. Yes, and it is a relatively small snapshot, and we'll have to see where we go.
Great. So I don't see any more questions coming in here. So if that is everything, I can bring this webinar to a close. I'd like to thank all of our speakers today for giving up their time to talk to us about their research. I think it's really important that we learn what's happening in the area of peat alternative research, and we can definitely learn from each other, I think. So this was really beneficial, I'm sure, for everyone. I'd like to acknowledge Donal Gernan as well, who is our mushroom advisor here at Teagasc, who is heavily involved in the organization of this webinar. And obviously, thank you to everyone for attending as well. And I hope everyone has a great day. So thank you, everyone.